Welcome to another episode of Inside the Future.
Glad to be here.
So imagine you have exactly 6 months to invent, test, and manufacture a completely new genetic drug, a drug that has never existed before anywhere.
Right, which is usually a decade-long process.
Exactly, a decade. But in this scenario, there's no blueprint, there's no precedent, and if you fail, a newborn baby dies.
The stakes literally could not be higher.
Yeah. They really couldn't. Today, we're taking a deep dive into a stack of recent sources. We've got papers from Science, Inside Precision Medicine, the Children's Hospital of Philadelphia, and UC Berkeley. And the mission for this deep dive is to uncover how scientists actually managed to pull this off.
It's a massive collaborative effort.
It is. We are looking at a bespoke, entirely customized CRISPR gene editing therapy designed from scratch for a single baby in just half a year. Okay. Let's unpack this.
To really understand the sheer velocity of that timeline, you have to look at the clinical crisis that set the clock ticking in the first place. The medical details here are, they're intense.
Yeah, reading through the sources, it's heartbreaking.
It centers on a baby boy named Kyle Muldoon Jr. He goes by KJ, born on August 1st, 2024. And by all initial clinical assessments, he appeared to be a perfectly healthy newborn.
Right. The parents are going through the normal routine, taking him home. Everything seems completely fine.
Yeah, perfectly normal.
But then, at just 2 days old, KJ's system essentially crashed. He became severely lethargic, he completely stopped taking in any food, and his core body temperature dropped to this really dangerous level.
The rapid decline, it immediately pointed the medical team toward a severe metabolic crisis. Specifically, something called carbamoyl phosphate synthetase 1 deficiency.
That is a mouthful, CPS1 deficiency, right?
Exactly, CPS1. It's an ultra-rare condition. We're talking about it affecting roughly 1 in 1.3 million births.
Which is incredibly rare. And knowing our audience, you likely understand the basic function of the liver as a filter, cleaning the blood.
Yeah.
But let's roll down into the molecular mechanics of this specific mutation. What exactly was failing in KJ's body?
Well, it comes down to a critical metabolic pathway called the urea cycle. So when your body breaks down proteins, it generates ammonia as a byproduct.
Like exhaust from an engine.
Right. Perfect analogy. Exhaust. And ammonia is highly neurotoxic. In a healthy system, the liver utilizes that CPS1 enzyme we mentioned to convert that toxic ammonia into urea.
Which is harmless. Right?
Yeah, harmless. And then it's just excreted in urine. But KJ was born with mutations in both copies of his CPS1 gene. His liver was completely missing that specific enzymatic machinery.
So he literally has no way to process the exhaust. The ammonia just builds up in his bloodstream.
And that buildup is catastrophic for the central nervous system.
Yeah.
A severe ammonia spike causes brain swelling.
Oh, wow.
Yeah, which can rapidly lead to irreversible neurological damage, coma, or even death. Just to give you an idea of the severity, statistically, only about 50% of infants born with severe CPS1 deficiency even survive early infancy.
That's terrifying. I mean, the clinical notes show they managed his immediate acute crisis by putting him on a highly strict, low-protein diet and administering these heavy nitrogen scavenging drugs.
Which is standard protocol for this.
Right. But looking at the literature, that is clearly just a frantic stopgap. I mean, they are constantly just bailing water out of a sinking boat.
That's exactly what it is.
Yeah.
Because the grim reality of the default long-term path for severe CPS1 deficiency is a full liver transplant.
Because you just need a new liver that actually has the genes.
Exactly. The only proven way to fix the missing enzyme is to introduce a completely new liver that has functional CPS1 genes.
But KJ was only days old. You can't perform a major organ transplant on an infant that small, can you?
No, the physics of the human body simply don't allow it. A newborn is just too fragile. Their vascular system is way too small to handle an adult or even a pediatric donor liver segment.
So what do?
They have to grow. They need to put on weight and stabilize medically to even survive the anesthesia, let alone the surgery itself.
Wow. So it's like telling someone they can have a life raft, but they have to tread water in a hurricane for a year before it arrives.
That is a very accurate way to put it.
Because while they are waiting for KJ to grow, he is living under the constant daily threat of an ammonia spike. I mean, if you consider normal childhood development, babies get sick.
All the time.
Right. If he catches a routine cold, his immune system fights it, cells break down, proteins are metabolized, ammonia is created, and he could suffer permanent brain damage before the new liver ever becomes a surgical option.
And the medical team at the Children's Hospital of Philadelphia, they recognized this impossible catch-22 immediately. The risk of permanent neurological damage during the waiting period was just unacceptably high.
So the standard of care wasn't going to work.
No, it wasn't going to save him intact,
No.
which forced them to pivot to something completely theoretical at the time. They realized they needed to repair his existing liver at the genetic level, and they had to do it fast.
And this brings us to Dr. Rebecca Ahrens-Nicholas from CHOP and Dr. Kiran Musunuru from Penn Medicine. Reviewing their research, they didn't just want to do, like, standard gene therapy. They aimed to build a customized CRISPR therapy tailored specifically to KJ's unique mutation.
The technological leap they took here is truly fascinating. When we talk about CRISPR, the dominant model most people know is CRISPR-Cas9.
The molecular scissors.
Right, the scissors. It cuts completely through both strands of the DNA double helix to disable a malfunctioning gene or to allow the cell to insert a new sequence during the repair process.
Which sounds a bit aggressive for a baby.
It is. For KJ, double-strand breaks can be chaotic. They are incredibly risky in a newborn's rapidly dividing liver cells. You don't want to shatter the DNA, so they used a much more advanced tool called a base editor.
I love how the source material describes this. Instead of taking scissors to the DNA, it's more like a chemical find and replace function in a word processor.
Yes.
Like they're just swapping a specific letter without deleting the rest of the document.
That is the perfect way to visualize it. A base editor only nicks one strand of the DNA to sort of open it up. It relies on a bespoke guide RNA, which is a sequence specifically coded to match the exact location of KJ's mutation on his CPS1 gene.
Okay, so it finds the exact typo.
Right. Once the guide docks at the correct location, an attached enzyme chemically converts one specific DNA letter into another, for instance, changing an A to a G. It corrects the typo without actually shattering the DNA strand.
But wait, I have to push back here. Isn't gene editing inherently risky? I mean, like trying to fix a single typo in a massive book, but accidentally deleting an entire paragraph somewhere else.
That's the big fear, yes.
Right, so if you put this into a living baby, how do you know the guide RNA won't mistakenly latch on to a similar-looking sequence on a completely different chromosome and, I don't know, mutate something vital?
You're touching on the single most critical hurdle in all of gen therapy off-target effects. You absolutely cannot just hope for the best. To mitigate this, the team partnered with the Innovative Genomics Institute at UC Berkeley. They ran the bespoke guide sequence through advanced computational models. These algorithms scan the entire 3 billion base pairs of the human genome.
3 billion.
Yeah, to predict any regions that look molecularly similar to the target site, where the editor might, mistakenly bind.
Okay, but looking closely at the methodology in the papers, there's a massive logical gap here that I want to ask about. The sources state they used animal models to ensure the medicine was safe before giving it to KJ. But if this guide sequence was engineered specifically and uniquely for KJ's personal human DNA mutation, how do you test that on a lab mouse? Mice don't naturally have KJ's exact genetic typo.
That is a very sharp deduction.
Yeah,
and it
right.
really goes to the heart of how regulatory safety testing has to adapt for personalized medicine, because you're actually testing two different things.
What do you?
First, you test the bespoke guide RNA on human cell lines in a lab. Often, these are cells engineered to replicate KJ's exact mutation.
Oh, I see.
That proves the chemical find and replace works. And you physically sequence the DNA afterward to prove there are no off-target edits in the human cells.
Okay, so the human cells prove the targeting is accurate. What do the mice prove?
The mice prove the delivery vehicle isn't toxic. You give the mice high doses of the lipid nanoparticles carrying a surrogate editor just to ensure the physical drug doesn't trigger a fatal immune response or cause liver failure. You are validating the safety of the delivery system in a living organism and the accuracy of the editing in human cells.
That makes total sense. And let's talk about that delivery system. You can't just inject raw genetic code into an arm and hope it knows where to go.
No, it would just degrade immediately. They utilized lipid nanoparticles, or LNPs. Those are essentially microscopic spheres of fat.
Like tiny fat bubbles.
Exactly. Inside these spheres, they packaged messenger RNA, mRNA, which carried the coded instructions for building the base editor.
And the choice of mRNA is deliberate, right? Because they don't want the editor to stick around in the body forever.
Exactly the opposite of, say, a viral vector, which might permanently integrate into the genome and raise the risk of cancer later in life.
Right.
The mRNA is transient. Once the LNPs are infused into the bloodstream, the liver, acting as the body's natural filter, rapidly absorbs these fat particles. The mRNA enters the liver cells read the instructions, build the base editor, fix the DNA, and then the mRNA naturally degrades and disappears within days.
So the editor is gone, but the genetic fix is permanent.
Precisely.
It's incredible. But developing a drug like this, designing the sequence, running the computational models, manufacturing the clinical-grade mRNA, doing the animal toxicity testing traditionally, doesn't that take an entire decade?
A decade is standard, yes, because the sequential nature of traditional pharma development is, well, it's incredibly risk-averse and bureaucratic.
But KJ didn't have a decade. Driven by the biological ticking clock of his condition and leveraging the massive manufacturing infrastructure of the Danaher Corporation alongside the IGI, this team compressed a 10-year drug development pipeline into 6 months.
6 months. It's unheard of.
From diagnosis to a finished clinical-grade vial of a drug that had never existed before. And when they took this mountain of safety data to the FDA turned around and approved the treatment for compassionate use in 1 week.
The sheer velocity of that regulatory turnaround reflects a deep paradigm shift.
Yeah.
I mean, the FDA recognized the catastrophic stakes, sure, but they also recognized the robustness of the computational and cellular safety data the team managed to compile.
Here's where it gets really interesting. Because while the biochemical sprint is breathtaking, we have to look at what this actually meant for the people living it.
Right, because the science is abstract until you place it in the context of a family facing an impossible choice.
Exactly. Imagine you are Kyle Nicole Muldoon. You've spent the first 7 months of your son's life practically living in the ICU at CHOP.
It's a nightmare scenario.
You are staring at monitors, terrified that if he gets fussy, it might mean his ammonia is spiking and he is suffering brain damage in real time. And then doctors offer you a choice. Wait for transplant that might never happen, or try an N-of-1 clinical trial.
And for those unfamiliar with the terminology, an N-of-1 trial means the entire clinical study consists of a single patient.
Just KJ.
Just KJ. The drug was built for him and him alone. It had never been tested in another human body.
Agreeing to an unproven, entirely novel genetic experiment on your newborn that requires a level of courage that is hard to fathom.
It really does. And Kyle Muldoon articulated the reasoning perfectly. He said, if you're going through something of this nature, it's important to find the tiniest bead of hope and hold on to that as long as you can.
Wow.
They understood the risks of the experimental therapy, but they also understood the absolute certainty of the traditional path. They wanted to give their son a chance to live outside the walls of a hospital.
So in late February of 2025, when KJ was about 7 months old, they administered the very first ANBI infusion of his bespoke therapy. They followed that up with a second dose in March and a third in April.
And the monitoring during those infusions was intense. With LNPs, there's always a risk of an inflammatory response.
Did he have one?
The clinical data shows there was a brief moment where KJ's liver enzyme spiked, indicating his immune system was reacting to the lipid nanoparticles.
That must have been terrifying.
I'm sure it was. But they managed it medically and the levels quickly stabilized. There were no severe adverse events.
And the results. After spending the first 307 days of his life inside the hospital, KJ was discharged. He went home to his siblings with a full police escort.
That's amazing.
The case studies note he is actively reaching his developmental milestones, like sitting upright unassisted. He is metabolizing higher levels of dietary protein, and his reliance on those heavy nitrogen scavenging drugs has plummeted.
But the true stress test of the therapy came a bit later. KJ caught a normal childhood virus, a rhinovirus.
Which previously would have been a life-threatening event.
Absolutely. The metabolic stress of fighting a cold would have caused a massive ammonia cascade. But post-treatment, his body weathered the viral infection normally. His ammonia levels remained stable.
Oh, that's huge.
It was the biological proof that the bespoke guide RNA had successfully edited his liver cells.
What's fascinating here is, looking at the clinical outcomes, the researchers are very careful to note that KJ is not entirely cured in a definitive zero-maintenance sense.
And that nuance is vital. The FDA protocol only permitted 3 doses to establish safety and efficacy. He still requires clinical monitoring and some dietary management.
Right, it wasn't a magic wand.
No, but the intervention successfully edited a sufficient percentage of his liver cells to completely alter the fatal trajectory of the disease. The primary objective, eliminating the need for a liver transplant and stopping the daily threat of brain damage, was achieved.
Which is life-changing.
And theoretically, as he grows and his liver expands, the platform allows for redosing if his enzyme levels ever drop.
It is a staggering victory for the Muldoon family. But reviewing the broader editorial published in Science by Dr. Peter Marks, the former director of the FDA's Center for Biologics Evaluation and Research, he makes it incredibly clear that this deep dive into KJ's story is fundamentally about the future of global medicine.
Dr. Marks framed KJ's case not as an anomaly, but as a proof of concept for how the medical establishment can begin tackling thousands of rare, previously untreatable genetic conditions.
Because if you look at the economics of modern pharmaceuticals, the industry is entirely built on scale. Companies spend billions developing a drug for millions of people with hypertension or diabetes to recoup their costs.
Exactly.
If a genetic typo only affects 1 in a million babies, the traditional economic model simply cannot justify the development cost.
Which is why they are classified as orphan diseases. Millions of patients globally suffer from rare conditions that are biologically understood but economically ignored. What Dr. Ahrens-Nicklas, Dr. Musunuru, and the consortium proved is the clinical viability of platform technologies.
So it's basically like building a universal, incredibly safe chassis for a car and then just swapping out the engine depending on the specific disease the patient has. You already know the chassis is safe and road tested, so you only have to verify the new engine.
That is the exact conceptual shift driving this new era. The lipid nanoparticles and the mRNA backbone that translates the base editor that is your universal chassis. It is standardized, it's stable, and its safety profile is increasingly well documented.
And the engine.
The only component that changes from patient to patient is the 20 nucleotide guide RNA of the engine.
And if you are the FDA, regulating a platform is entirely different than regulating a novel drug from scratch every single time.
The regulatory implications are massive. Instead of forcing scientists to conduct 10 years of phase 1, phase 2, and phase 3 trials for every single rare disease, the FDA can evaluate the platform as a whole.
That changes everything.
It does. If the delivery mechanism is...
And the sources highlight that this is already moving beyond theory. The IGI and Danaher Corporation have officially partnered on the Beacon for CRISPR Cures project. Their stated mandate is to create a scalable, affordable cookbook approach to on-demand gene editing.
They are targeting a wide spectrum of conditions. For instance, they are actively working on Artemis SCID, which is a devastating rare genetic disorder where infants are born without a functioning immune system.
Often historically referred to as bubble boy disease, right?
Yes, exactly. The Beacon project aims to take the exact same platform architecture used for KJ, swap the guide RNA to target the Artemis mutation, and deploy it.
So what does this all mean? We started with a newborn in a catastrophic metabolic crisis, explored a frantic 6-month scientific sprint to rewrite his biological code, and now we are staring at a scalable cookbook for curing orphan diseases globally.
If we connect this to the bigger picture, Dr. Musunuru's ultimate vision is to fundamentally change the standard of care. Right now, this 6-month sprint was a heroic, exhausting effort requiring the combined resources of multiple massive institutions.
Yeah, they can't do that for every single baby.
Right. But the goal is normalization. In the future, if a child is born with an inborn error of metabolism, the clinical response won't be panic or an organ transplant waitlist.
What will it be?
Doctors will simply pull the established LNP chassis off the shelf, synthesize the specific guide RNA for that child's mutation, and correct the error in their liver before the disease ever has a chance to manifest symptoms.
It transforms a bespoke medical miracle into a routine outpatient procedure. It democratizes access to precision medicine for diseases that the market previously ignored.
And it proves that the bottleneck is no longer our understanding of the biology, but our ability to streamline the manufacturing and the regulatory oversight of these platform technologies.
When you consider the sheer speed at which this field is moving, it leaves you with something profound to mull over. Imagine you're facing this kind of diagnosis in the future, or someone you love is. What happens to our very definition of a fatal disease when a bespoke, customized genetic cure can be coded, manufactured, and delivered to your body in the time it takes to process a passport application? How will this new era of on-demand biology reshape how you think about your own genetic destiny?
It's a question we'll all be facing sooner than we think.
Thank you to all our listeners, and stay tuned for the next episode.